Good evening, Ayman. Good evening, Vanshika. Okay, so today we'll be dealing with the first part of the oogenesis that we had commenced last time. So we'll be dealing with it very conceptually so that your entire NEET and board exam can be taken into consideration. Okay, so some things are there which are really not explained till the, you can say, conceptual level in the NCRT examination book, but they have been, you can say, the words have been put in the NCRT book of those things. Like you have the antrum, you have the follicle. Okay, so those things are described in detail, and also the diplotene arrest that you might be knowing. Okay, the diplotene arrest happens when you have taken birth, when you have just taken birth, but it gets broken when you have attained puberty as a female. Now, why does diplotene arrest happen and how does it get broken? Okay, these two things will also be answered. So, these are the questions that they have put in NCRT, but the direct answer is not given in the book. Okay, the thing is, so let's begin the discussion. So, when we are born, okay, regarding oogenesis, when we are born, what we have is we have a supply of 2 million primary oocytes. Okay, 2 million primary oocytes are present. Now, what happens is, after that, what happens is, what we have is, till we reach the stage of puberty, okay, till we reach the stage of puberty, only 40,000 only 40,000 of the primary oocytes they are able to survive. That's a great dip in the number, and only they are arrested in the prophase 1's diplotene stage. Okay. Now, this is not the end of the story. What happens is, from this number, which is the 40,000, approximately, you can say, if you have 400 menstrual cycles throughout your life, so approximately 400, that is one per menstrual cycle, are only ovulated. So out of the 40,000, only 400 are ovulated. Considering the average number of menstrual cycles, okay, that you may have is 400. Okay, so only 400, one per menstrual cycle, are ovulated. The rest of the primary oocytes, what happens to them is they undergo degeneration without leaving the ovary. Okay, some of them can proceed in the growth. Okay, can proceed in the growth, but they do undergo degeneration. That degeneration process is known as atresia. Okay, atresia is asked in NEET examination. That is nothing but degeneration of the primary oocytes. Now, when you have degenerated, they are supposed to known as they have become atretic. Okay, so what happens is, some studies do, you can say, suggest that the adult mammalian ovaries contain also primitive cells, okay, that can give rise to new oocytes, but that is contradictory. That is why the females, they attain menopause. Okay, what happens in males is, when the male cells undergo, or the male spermatocytes, or spermatogonia, I should be saying, they undergo spermatogenesis. What happens is, the type A spermatogonia, they differentiate into, it undergoes mitosis, it becomes type A again, and then type B. This type B spermatogonia then undergoes spermatogenesis. Okay, so what happens is, the type A spermatogonia has stem cell like property. That is why males are able to always, throughout their life, they are able to produce sperms. Okay, but females, they cannot ovulate throughout their life, because some studies do suggest that they may be having the primitive stem cells, but they're still controversial, meaning that till now there is no study that has explained that females can have sperm, you can say, stem cells. So directly the oogonium will take the path of oogenesis, and this extra mitosis doesn't happen, so that the type A here, like the type A cells, their number are being, you can say, they stay constant. Their number, their specific number are being maintained. They are not getting depleted. So this doesn't happen in female. There, this type A directly will enter into spermato, you can say, oogenesis, and therefore the number of primary oocytes will be directly reducing after each and every oogenetic event, right? So that is why the females, they undergo something known as meno something known as menopause, but males, they do not undergo menopause whatsoever. But we don't have any menstrual cycle, but we do not undergo, okay, we do not undergo, you can say, spermopause, like you can say. We can produce sperms if we are healthy. Many unhealthy men, they cannot produce, but if you are healthy till 80, 90 years old also, we can technically make any, you can say, lady pregnant via our sperms. We can produce sperms till our death. Biologically males can do that, but females, they cannot ovulate after a certain period of time, that is 45 to, you can say, 55 years of age, when they attain menopause. That's the thing, depending on the sperm cell regeneration event which happens in males, but that doesn't happen in females. Okay, now the egg or the ovum along with its surrounding cells is known as a follicle. So suppose this is an egg. Okay, so suppose this is an egg and this is the surrounding cell. So suppose this is the surrounding cell. So this will be known as a follicle. Okay, now this is known as a follicle. Now, what will happen now? The maturation of the egg, the follicle will start becoming mature. Now, the maturation of the egg, that specific table that I had given to you in the last class, you can refer to that table. The maturation of the egg is intimately bound with the development of its cellular covering. Okay, always remember this thing, that the cellular covering over the egg, the number of, you can say, follicular cells, they are the granulosa cells. They will decide which type of ovary you are, whether you are a primary oocyte. They will tell you which type of oocyte you are. Okay, which type of follicle you are. That will be decided by them, whether you are a primary follicle, a secondary follicle, tertiary follicle slash, you can say, Graafian follicle, and so on. Okay, so what happens is, the growth period of a human oocyte, it can last between anything from 110 to 120 days. 110 to 120 days. This is a neat question. What is the growth period for a human oocyte? Okay, during its period of growth and maturation, what happens is, the oocyte must accumulate, means must be able to synthesize a lot of, you can say, organelles, rRNAs, okay, mRNAs, tRNAs, to carry the embryo through the first few weeks or days, first few days of its existence, meaning that it should generate, okay, organelles, then mRNA, a lot of mRNA, okay, tRNA, a lot of rRNA, because if you don't have a lot of rRNA, where will the mRNA go and bind? If you don't have a lot of mRNA, from where will the protein be synthesized? If you don't have tRNA, who will be charged? The tRNAs will be charged with the amino acid. Okay, this entire process, the translation process, if you remember properly, is dependent on this specific event, this specific concentration of rRNA, tRNA, and mRNA. Okay, the most prolific is the rRNA. Okay, so the ribosomal RNA, so they must amass or accumulate so many organelles. Okay, so that they should be having, good evening Asma, so they should be having so many, you can say, organelles and rRNA, so that they can sustain the embryo just after the fertilization event for few days. Okay, so that is what the entire scenario is of a human oocyte. So in the embryo, when we talk about the embryonic, you can say, development in the embryo, first thing, objective question, the oogonia are always naked. Okay, The oogonia are always naked in the embryo, means no follicular or granulosa cells are present on top of it. But after the meiosis begins, okay, cells from the ovary, they partially surround the primary oocytes to form primordial follicle. Now some cells will start surrounding it and it will become primordial oocyte. Okay, primordial oocyte in case of it being just before the birth, it will become primordial oocyte in the fetus. Now by birth, the primary oocytes are completely invested with a, you can say, complete layer of follicular cells, which are the granulosa cells. Okay, and it then becomes a primary follicle. It then becomes a primary follicle. This is the oocyte. Okay, now this will be invested with a full layer, a complete layer of, you can say, granulosa cells. These are the granulosa slash follicular cells. Now you will be wondering why I haven't made them perfectly in contact with this, because now there will something will be discussed very important from the board and competitive exam, both the perspectives, known as zona pellucida. Okay, because while the primary follicle takes shape, a prominent translucent non-cellular membrane known as the zona pellucida forms between the primary oocyte and its enveloping follicular cells. So I'll write this down. A prominent translucent and non-cellular is the most important. Okay, a prominent translucent non-cellular, or it is also known as acellular. Okay, acellular, I can say much better, acellular membrane called as the zona pellucida. Zona pellucida forms between the primary oocyte between the primary oocyte, okay, and the surrounding follicular or granulosa cells, and the surrounding follicular slash granulosa cells, meaning that if you see, you can draw this layer here. Okay, you can draw this layer here. This is your zona pellucida. Now this layer is of great importance. Why? This layer is of great importance because it is just not an acellular layer which has no importance. In biology, everything has what is specific reason behind it. Okay. So the zona pellucida, always know, is made up of glycoproteins and also glycosaminoglycans. Very good competitive exam question. So this zona pellucida contains glycoproteins, made up of glycoproteins and glycosaminoglycans. Now, glycoprotein is proteins which have been directly modified by covalent linkage of glucose residue, either N-linked or O-linked, via the transferase enzymes present in the Golgi apparatus. That is post-translational modification. Glycosaminoglycans are something different. They are specific sugars, especially the disaccharides, okay, which have sulfur group and also amino group. Okay, so the example of this is, you might have heard of heparan sulfate, okay, dermatan sulfate, okay, then chondroitin, okay, sulfate, then hyaluronic acid. So these are present in our extracellular matrix outside the cell, okay, present in the synovial fluid also, okay. So they are also present in zona pellucida, and they also have specific receptors. They also have specific receptors like ZP1, ZP2, ZP3, zona pellucida 1, 2, 3, which will bind to what? Which will bind to the sperms. Very, very important. They have specific sperm binding receptors as well. Okay, the zona pellucida, they have specific sperm binding receptor as well. They bind to the zona pellucida layer. Okay, so the thing is, in you can say rodents, mice and all, it has been seen that the components of the zona pellucida, that is the glycoprotein and glycosaminoglycan, they are synthesized almost entirely by the primary oocyte or egg only. But in other mammals like us, the follicular cells, meaning this granulosa cell and egg, they both contributes towards the synthesis of zona pellucida. Okay, so very, very good, you can say, a very, very important difference between mice and humans, you can say. Now, secondly, what I want to show you is, I want to tell you something known as a specific event. Okay, I'll just share my screen with you. Okay, I'll just share my screen and show you a very, very, you can say, great diagram. Okay, which you will really love the diagram. So the diagram is, so I'll just share the screen regarding the diplotene arrest now. Okay, regarding the diplotene arrest, I'll share the screen and show you a very good diagram. So just tell me, okay, I'll just share it right now. You'll be able to see the diagram in 2 seconds. Okay, just hold on, I'm sharing the diagram. Now you'll be able to see this diagram. Okay, so I think you are able to see the diagram right now. Okay, so I think you are able to see the diagram, right? So the thing is, just look at this diagram. Okay, just look at this diagram and you will be able to comprehend what I am trying to say. Okay, so what I am trying to say here is, just look at this diagram. You have everything here. Okay, so what I'm trying to say is, the oocyte and its surrounding follicular cells, okay, they maintain an intimate structural and functional relationship through two types of transzonal projections. Okay, is given in this diagram. One is perpetuated by the F actin, like actin induced filamentous actin, that is you might have learned in class 11th, right? The other one is the M transzonal protein, that is covered by the, or that is directly perpetuated by the, you can say, microtubules. Okay, so they form the adherence junctions, and through which the transportation of specific substances from the follicular cells or the granulosa cells to the oocytes can take place. Now, the meiotic arrest that we talk about is, the meiotic arrest, you can see, at the diplotene stage of the first meiotic division, is the result of a specific pathway. Okay, the specific pathway is the principal factor known as a specific molecule by the name cyclic adenosine monophosphate, CAMP. You can look here, CAMP. Okay, you can look here, theca interna, theca externa, everything will be there. You can see here, all those things are there. So you have the zona pellucida here, you can see the membrana granulosa. So all these things I'll explain it to you. Okay, so the thing is, first what happens is, you have something known as cyclic AMP. Now what happens is, the production of cyclic AMP happens in the oocyte. Okay, and also it occurs in the follicular cells. Okay, So it happens in the oocyte and it happens in the also in the follicular cells. Now, its transport happens into the oocyte through the gap junctions that connect the follicular cells to the oocyte. Okay, that connects the follicular cells, these follicular cells to the oocyte. Okay, you'll be having the selectin proteins, the integrins, okay, the cadherins, all types of proteins you will be finding in the gap junction. Okay, so the thing is, then what you will be finding is you will be having this kind of gap junctions and the transportation of the CAMP will take place. Once the transportation of CAMP takes place, okay, so I'll just switch it and explain the pathway to you. Once the transportation, so what happens is we have here, you can say the zona pellucida, it can be drawn like this. Okay, then I can draw this follicular cells like this. Okay. So now what will happen is they will produce a molecule. This will produce a molecule known as CAMP. Okay, cyclic adenosine monophosphate. They will produce cyclic adenosine monophosphate, and this cyclic adenosine monophosphate will be traveling through the gap junctions into the primary oocyte. Okay. Now, what will it do is this cyclic adenosine monophosphate will travel also in addition to CAMP. The follicular cells also produce and transport another molecule known as CGMP. Okay, they transport another molecule known as cyclic guanosine monophosphate (CGMP). And what happens is when you can say the CGMP reaches the oocyte, it will inhibit a specific enzyme known as the CGMP inhibits, you can say, PDE3A. Okay, PDE3A. PDE3A is nothing but phosphodiesterase 3A. This PD3A does what? PDE3A does what? It converts the cyclic AMP to 5 prime AMP, means it will inactivate the cyclic AMP. Now what happens is this cyclic AMP is responsible for inhibiting MPF, mitosis promoting factor, okay, or maturation promoting factor, sometimes it is known as meiosis promoting factor or maturation, mostly maturation promoting factor. So if this is inhibited, the cell stays arrested in the diplotene stage. So this is how the diplotene arrest is maintained. This is how the diplotene arrest is maintained. Okay, this is how the diplotene arrest is completely maintained. Okay, now what happens is after this diplotene arrest is maintained, what you get is this diplotene arrest will keep the primary oocyte arrested in the diplotene stage till you attain something known as puberty. Now everybody has heard of puberty. Now in the pre-pubertal years, means before puberty, I'll share that diagram again with you. So in the pre-pubertal years, so what happens is you will look at the diagram. Many of the primary follicles they'll enlarge, as you can see in the diagram. Mainly why? Because of an increase in the size of oocyte. The oocyte increases as much as 100 fold. Okay, the oocyte increases as much as by 100 fold. Okay, and also the number of follicular cells or the granulosa cells, they also keep on increasing. Now, an oocyte with more than one layer of surrounding granulosa cells or follicular cells is known as a secondary follicle. Okay, you can just look at this image, is known as a secondary follicle, more than one layer. So you can have, see here, more than one layer are present of follicular cells. So they are known as the secondary follicle. Okay, now what happens is, there then after the secondary follicle is produced, a basement membrane is also produced. Okay, a basement membrane, this one, if I zoom in this image, okay, you will see this is the basement membrane. Okay, this one in the violet, known as membrana granulosa. This is the basement membrane which is produced. Basement membrane connects, always know this, granulosa cells are epithelial cells in nature, and basement membrane connects epithelial to connective tissue. They are made up of, you can say, the basal lamina and the reticular lamina. If you have studied the basal membrane in class 11, okay, so they are made up of basal lamina and reticular lamina. That is, the basal membrane is a membrane which connects the epithelial to connective tissue. Always remember. So the basal membrane here is the membrana granulosa. Means after this, what will be there? Connective tissue. Since these are epithelial, granulosa cells or follicular cells are epithelial cells in nature, then there has to be these cells, okay, which is known as the developing theca here. You will be having theca interna, externa also, I'll explain to you. Those will be the connective tissue cells. Okay, now this membrana granulosa surrounds the epithelial granulosa cells of the secondary follicle. Now the membrana granulosa cells, okay, or the membrana granulosa basal membrane, it forms a barrier to capillaries. Okay, and as a result, the oocyte and the granulosa cells, they depend on diffusion of oxygen and nutrients for their survival. Very, very important thing this is, that this membrana granulosa is it does form a barrier to capillaries, because it is like this. So no, now the capillaries which are directly transferring nutrients cannot. Now the oxygen and nutrients will diffuse through this basal membrane. Now directly capillaries cannot penetrate this entire follicular cells. Okay, so this membrana granulosa, they form a barrier to the capillaries that will then cause the diffusion of oxygen and nutrients which will be needed by these cells. Otherwise they cannot produce ATP, right? Without nutrients they cannot have any activating enzymes inside the cell. So you need oxygen, you need nutrients, because oxygen is the last electron acceptor in the electron transport chain. So that will occur through this basement membrane, this membrana granulosa, only via diffusion process now, because the capillaries cannot penetrate the membrana granulosa. Okay, get that very, very clearly. Now what can happen is, the membrana granulosa has formed the barrier. Now an additional covering will be produced, that you are looking at the thecal layer. After that, they are derived, the additional, you can say, cellular covering, they are derived from the ovarian connective tissue, which is known as the stroma. The ovarian connective tissue is known as stroma, you can say. Okay, the ovarian connective tissue is known as the stroma. So from the connective tissue, these thecal cells have been produced. So these thecal cells are the connective tissue. Okay, so they develop after it has become at least 2 to 3 cell layers thick. Okay, so initially, okay, they are known as theca, developing theca, folliculi theca. Okay, but later on it differentiates into two layers. The two layers are theca interna and theca externa, that you are looking here. Theca interna, internal, which is attached to the membrana granulosa, and theca externa. Theca interna, if you see, it is highly vascularized, means a lot of blood vessels are present in theca interna compared to the theca externa. Okay, Theca interna is highly vascularized. Now, how they are vascularized? Because they will be secreting angiogenetic factor, means they which will promote the formation of blood vessels, okay, like VEGF, vascular endothelial growth factor. So those kinds of proteins will be secreted to produce more and more, to cause the production and synthesis of more and more blood vessels, okay. Now, early development of follicle occurs without significant influence of hormones like follicle stimulating hormone, okay. So early development, it happens, you can say, I'll write it down. So we have here theca interna, which is highly vascularized, okay. Theca externa, which is less vascularized, means less blood vessels. They both are derived from the stroma, that is the connective tissue of the ovary. These 1 mark questions, they come a lot, okay, especially in the competitive examination. Connective tissue of the ovary. Also, the thing that I told you, this one, if you draw, okay, this is the ovary, this is the, you can see, zona pellucida, and suppose this one is the granulosa cells, layer number 1, this is suppose layer number 2, this is layer number 2. Now you will be having, you can say, this is known as membrana granulosa. This is the basement membrane, okay, known as membrana granulosa, okay. So now this will connect, since these are the granulosa cells or the follicular cells, they are epithelial in nature, okay. Now they will be connected to, you can say, thecal cells. These are the theca interna, you can say. These are the theca interna, and these are the theca externa, you can say. You can draw bigger ones. These are the theca externa. They're all derived from the stroma, so they are all connective tissue. This is theca interna. So that is the way the follicles they develop. Okay. So this is the development of the follicle. Okay. This is, and this membrana granulosa has stopped the in growth of the capillaries. So now oxygen, okay, O2 nutrients will all go via, you can say, O2 nutrients will all go through the membrana granulosa into the follicular cells and oocyte, okay, primary oocyte, by only diffusion process. Always remember, by only the process of simple diffusion. Now, after this has happened, what happens is FSH doesn't affect the early follicular growth. Okay. FSH, follicular stimulating hormone secreted by the anterior pituitary gland. Okay, it is known as adenohypophysis. You might be knowing from class 11th endocrinology chapter. So it doesn't affect early follicular growth. Doesn't affect early follicular growth whatsoever. Okay, but what happens is it will affect the later follicular growth. Now, how will it affect the later follicular growth? What happens is the follicular, by the time you can say puberty approaches, okay, continued follicular maturation, it will require the action of the follicular or the gonadotropic hormone, which is known as the follicular stimulating hormone FSH. And FSH will act on what? Since the follicles are developing, secondary follicles has produced, the FSH will act on granulosa cells. Now, by this time, the granulosa cells, these cells have developed receptor for FSH. They have developed receptor for FSH. Now, FSH can act on the granulosa cells. Okay. Now, since you can say FSH is acting on granulosa cells, so the effect will be, you can say, the most obvious indication will be some of the follicles will show the formation of an antrum. Antrum is nothing but a cavity filled with fluid, and that fluid is known as liquor folliculi. I'll again show the screen to you. That same screen that I was sharing. Okay. Now look what does. Now you can see, I think, the image. Yeah. So this is what it does. This is now the antrum. Okay. Go down. This is the antrum. Now, because of this FSH, the follicular cells have been able to secrete a fluid. Now, the fluid is present inside the follicle. Now, that fluid filled cavity is known as antrum, and this fluid is known as liquor folliculi. Okay, this fluid is known as liquor folliculi. Okay, always remember. Now, you can see here two types of cells. Okay, cumulus oophorus or cumulus cells and mural granulosa cells. The cells that immediately surround the oocyte. Okay, you can see here the cells that are immediately surrounding the oocyte, direct contact, they are known as cumulus oophorus or cumulus cells, and the cells which are not in direct contact, this one, the follicular cells only. Okay, they are talking about the follicular cells only. But the follicular cells which are directly, or the granulosa cells, the epithelial cells which are directly surrounding the oocyte are known as cumulus oophorus or simply cumulus cells, and the follicular cells which are not in direct contact, okay, with the granulosa cells, which are present between the antrum and the membrana granulosa, okay, that is known as mural granulosa cells, okay, because they are present between the antrum, you can say, and the membrana granulosa. So they are demarcating membrana granulosa from antrum. So they are known as mural granulosa cells. Very, very important. Next thing what I'll tell you is, now what happens is there are factors secreted by oocyte which confer different properties on the cumulus cells from the mural cells. So they are a little bit different. Okay, there are various factors which are not in your course. So mural cells are different from the cumulus cells. Don't think that just because of the name it has been demarcated. There are changes in the function as well. Okay. Now the thing is, in the absence of a direct stimulus from the oocyte, the granulosa cells or the simple granulosa cells, they will follow a default pathway and you can say assemble the FSH receptors on their surface. But in contrast, the cumulus cells, they do not express any FSH receptor. Okay, very, very important. The cumulus cells, they do not express any FSH receptors. Okay, so what happens is the enlargement, you can see the enlargement of the follicle, it results largely from the proliferation of granulosa cells. Now what happens is when it responds to the, you can say, FSH, okay, it responds to the FSH, what will happen is the cells of the theca interna, okay, when we talk about theca interna and externa, so cells of the theca interna, what you are seeing in this image, this theca interna cells, okay, they possess the receptor for luteinizing hormone. Very, very important. FSH receptors are present in the granulosa cells. LH receptors are present on the theca interna cells. So they possess the receptor for the luteinizing hormone. Now what happens is the theca interna cells, okay, what it does is they will, you can say, it produces testosterone, you can say a variant of testosterone after they have been stimulated by luteinizing hormone. Okay, so the thing is, I'll just explain it to you. You can just see here. It is when LH binds with the, you can say, LH receptor on the thecal internal cell, it produces testosterone T. This testosterone passes through the membrana granulosa cells, okay, passes through the membrana granulosa cells. Just look here, okay, and membrana granulosa cells, since they are expressing FSH receptor, so in the influence of FSH they'll make or release an or synthesize an enzyme, a protein known as aromatase. Okay, they'll synthesize this protein aromatase, and this aromatase converts testosterone to estrogen. Okay, that is 17 beta estradiol, simply known as estrogen. Now not only does this estrogen, now they leaves the follicle and will exert its effects to the other body, making secondary female characteristics come. Breast development, then lesser hair on the body, you can say less hoarser voice, more hair, okay, all those things. But it also stimulates the formation of LH receptor now on the granulosa cells. Okay. Now through this mechanism, the follicular cells are able to respond to the LH surge that I'll be teaching you in the menstrual cycle. Now this estrogen will act on the granulosa cells and will cause the expression of LH receptor on the granulosa cells also. Okay. So what happens is, now the follicle enlarges and now it becomes the tertiary follicle, also known as Graafian follicle. So the resumption of meiosis, how will it happen? To the LS cells is stimulated by the granulosa cells. Okay, because the oocyte doesn't possess any LH receptor. So the cumulus cells, while LH binds to the cumulus cells will shut down their gap junction. Okay, when they shut down their gap junction, CGMP cannot enter, okay, to the oocyte, and then you can say entire event will be reversed. Now MPF will not be inhibited. The thing is, this is what happens. Okay, I'll write it down little bit, explain it in, you can say, on the whiteboard also. So just look here. So what happens is the granulosa cells, okay, the granulosa cells, these ones, they only express FSH receptor, okay, they express FSH receptor only, whereas the thecal cells, okay, they express LH receptor. And when LH comes and binds, they secrete testosterone, okay. Testosterone goes through the membrana granulosa and comes into the granulosa cells, and the granulosa cells will release a hormone, sorry, an enzyme, which is a protein known as aromatase. Okay, due to the influence of FSH will cause the granulosa cells to release the enzyme aromatase, which converts testosterone into 17 beta estradiol, okay, which is nothing but a type of estrogen. Now this estrogen, okay, will act on the same granulosa cells, and will cause the granulosa cells to express LH receptor now. Until now, they were not expressing LH receptor, but now due to the effect of estrogen, they will express LH receptor, and this estrogen will also go to the other body parts and cause secondary sexual character development. Okay. Now, due to it expressing LH receptor, now the granulosa cells will bind to luteinizing hormone, and when it does bind to luteinizing hormone, what will happen is the cumulus cells mainly, they will close their gap junction. They will close their gap junction. Okay. Now, when they have closed their gap junction, what will now happen? Now, suppose this is the oocyte, this is the zona pellucida, and the gap junctions have been closed. So I told you that the cGMP and CMP, mostly the cGMP which was coming in, cyclic GMP, now this cannot come in, or it cannot reach oocyte. Now I told you this cGMP was inhibiting, you can say, PDE3A. Now if it cannot reach, means now PDE3 will be active. When PDE3A will be active, the cyclic AMP, which is produced by the oocyte only, will be degraded by phosphodiesterase 3A to 5' adenosine monophosphate. Now since this is broken, so now cyclic AMP was inhibiting maturation promoting factor. Now since this has become like this, so this will not be inhibited, this will be now active. And now what will happen? The diplotene arrest will be broken. Diplotene arrest broken. Until that time, this follicle would have become tertiary follicle. Tertiary follicle, also known as the Graafian follicle. So this is the event why, and this is the, you can say, reason why the diplotene arrest is broken at puberty, and this is what Graafian follicle is all about. This is what your Graafian follicle is all about. So this is how you can learn about, this is how you can understand what is really happening in your body. Okay, just not that diplotene arrest happens, it gets broken. This is how it happens. After birth it stays, and this is how it gets broken. So the LH receptors are expressed on the granulosa cells when and only when the estrogen is produced. How will estrogen be produced? When FSH receptors are produced on granulosa cells, the granulosa cells will secrete aromatase. Now aromatase will convert testosterone. From where does testosterone come? Because the theca interna cells will express the LH receptor. Okay, so you can write it down like this. LH receptor first expressed on theca interna cells. You can write a flowchart thing. Then theca interna cells will do what? They will secrete testosterone hormone. Next, you can say testosterone will pass through the basement membrane known as membrana granulosa. Then it will reach granulosa cells, and ganosa cells already express FSH receptor. Now FSH acting on FSH receptor on granulosa cells will cause the granulosa cells to release aromatase enzyme. Now testosterone has reached granulosa cells and FSH is causing granulosa cells to release aromatase. Now the reaction will be testosterone will be converted to 17 beta estradiol, which is nothing but estrogen. Now this estrogen will leave the follicle and cause secondary sexual development in the other body parts and also induce the granulosa cells to express LH receptor. Okay, and when now LH reacts with the or binds with the LH receptor on the granulosa cells, then ovulation will take place and also gap junctions will close, and then only ovulation will take place because diplotene arrest will be broken. Diplotene arrest is broken, and then ovulation, because just before ovulation we have LH surge in the menstrual cycle, right? So this is how the entire thing is perpetuated. This is the entire event pertaining to oogenesis. Okay, so after this what will happen is ovulation has happened. Okay, and then in during the ovulation process the oogenesis has, or the primary oocyte has become secondary oocyte. Okay, so it has become secondary oocyte, and a first polar body has been generated, polar body 1, and the secondary oocyte enters meiosis part 2. It gets arrested in the metaphase, that is the metaphase 2 of meiosis. Okay, and until this secondary oocyte which is arrested in metaphase stage gets fertilized by the sperm, it cannot complete its complete meiotic event. If it gets fertilized, if fertilization occurs, means sperm entry via the acrosomal reaction, which will be taught later on. Okay, then the secondary oocyte overcomes you can say overcomes metaphase to arrest, and then produces, you can say directly it will produce the fertilized ovum, also known as oocyte plus polar body 2. Both the polar bodies, they degenerate, okay, by an unknown mechanism. Still research is going on. Why does the polar body degenerate and how does it degenerate? But both the polar bodies degenerate. So this completes your entire process of oogenesis. If you have any clarifications regarding this, you can really ask me on my personal number. I'll be replying as soon as possible. And thanks a lot. Now let's meet in the next class and discuss spermatogenesis and the male and female reproductive system also in detail. Okay, then thanks a lot.